Carbon allocation to mycorrhizal fungi and nitrogen uptake in a boreal forest. Nearly all land plants have a relationship with soil fungi called mycorrhizae, which play a key role in shaping evolution, ecology, and community structure of terrestrial plants. This is essentially a symbiotic relationship that ensures that fungi receive sugar from the plant and then sends back nutrients and water through the roots. So plants acquire carbon from photosynthesis, then exchange it for nutrients gained from an extensive underground network of mycorrhizal hyphae. In a lot of ecosystems where nitrogen limits plant growth, tree roots are covered in ectomycorrhizal fungi to help absorb nitrogen. The goal of this study is to determine the effects of changes in carbon and nitrogen supply on the transfer of nitrogen between EM fungi and its host trees. To do so, the scientists installed tree shading treatments designed to reduce carbon supply to plants and 3 levels of nitrogen applications to investigate how different amounts of carbon and nitrogen will affect the transfer of nitrogen. This study was conducted in a relatively young Swedish forest of Scots pine, about 175 meters above sea level. The experimental design consisted of 18 different 10 by 10 meter plots assigned different shading and nitrogen treatments. Nitrogen treatments consisted of adding a nitrate tracer, which was a nitrogen-based fertilizer at a low rate and a high rate. The low rate was 20 kilograms of nitrogen per hectare, and the high rate was 150 kilograms of nitrogen per hectare. And then each was compared to unfertilized control plots. The shaded treatments were done to emulate reduced carbon intake, so half the plots were shaded with a nylon mesh. Measurements were collected after 1 week, 2 weeks, and approximately 4 weeks after the nitrogen treatment was applied. The scientists collected soil respiration measurements using a portable gas exchange system. They also collected fungal chitin, pine needles from each plot, and sporocarps from each plot. All this matter was dried and then tested for nitrogen and carbon content using an elemental analyzer. The scientists found 4 key results. 1, that shade treatments worked. They did exactly what they needed to do, which was reduce carbon intakes from the plants. Next, they found that nitrogen treatment was linked to a higher nitrogen concentration in the pine leaves. Third, they found that shading or carbon reduction caused greater nitrogen content in tree foliage as well. And lastly, they found that unshaded regions are more likely to show greater nitrogen content in fungal biomass. In conclusion, we clearly see that altered carbon and nitrogen have an effect on fungal and plant relationships. We can conclude that greater allocation of carbon to EM fungi results in reduced nitrogen transfer to its host trees. Higher nitrogen concentrations in EM fungal biomass and tree foliage in shaded regions support the theory that when more carbon is sent to mycorrhizal fungi, less nitrogen is transferred to the host trees, highlighting the nitrogen limitation for tree growth. As summer cloud cover increases, allocation of carbon will likely be reduced to mycorrhizal fungi over the long term. Under these weather conditions, along with greater volumes of nitrogen deposits associated with human activity, both fungal and plant demand for nitrogen increases, so there's a greater potential to lose nitrogen from the ecosystem. From this study, we see the need to involve mycorrhizal dynamics into forest ecology to better understand the relationship between carbon and nitrogen in our ecosystems. We also see that carbon and nitrogen deposits have profound effects on forest growth, and the need to protect these lands promote the storage and retention of these elements within our forests. These discoveries will allow us to better understand how intensified human activity affects the biochemistry of our forested ecosystems.